Okay. Have you guys heard the term orthologous regions versus paralogous region? Some of you have, right? And when we are doing phylogenetic analysis, we have to make sure that we use orthologs, right? And this is a very important point, okay? So what is homology? Yeah, similarity because of common ancestry, right? So people also use the term homologous genes or homologous regions, which basically means that, you know, these genes share a common ancestor, right? From that common ancestor, these genes have been derived. And one should actually use homologs when we are, gene homologs when we are building phylogenies, right? So this common ancestor of A, B, C, and D had one particular gene. That gene was then inherited by the daughter species. Today you sequence that gene. They are pretty much identical. They are pretty much identical because they were all derived from that one common ancestor that had the gene. And when you build that phylogeny, you build a phylogeny using that gene, it retrieves the actual species phylogeny, right? So genes derived from a common ancestor. So similarity between two genes or similarity between any two regions in different species can be attributed to homology. They have been inherited from the same common ancestor. However, you can have gene duplication event, and which is very, very common. Right? So in this common ancestor, there was a duplication event, and now you have a copy of gene 1, let's call that gene 2. This is pretty much a different gene for all practical purposes. And now the daughter species have all inherited those two genes. If you compare the same copy in the daughter genes, we call it orthologs. Okay, so you're comparing the same copy. They are orthologs. But if you are comparing across copies, we call them paralogs. Right? So genes that are homologous due to speciation, so these are homologous because of speciation, they are called orthologs. And genes that are homologs due to gene duplication event are called paralogs. Does that make sense? Any questions? Another way of looking at it is that you have to use orthologs in your phylogeny. Don't use, in some species this gene, in other species that gene, then you'll get a completely messed up phylogeny. Because it is almost like using another gene. Okay? Now, if you just use gene 1, you'll get phylogeny like so, gene 2, another phylogeny, perfect, everything is under control. Now, if you use both of them, both the copies, in an alignment and build a tree, the tree will look like this. Gene 1 will retrieve the species phylogeny, gene 2 will retrieve the species phylogeny, and they both are related to each other because of a gene duplication event. Right? So this is how it will look like. So it will be like, you know, you expect just one tip, right? Instead you have like, for each species, there's a mirror image in the other gene. Okay. The complication arises when you include a paralog in your alignment. Okay. So here there was a gene duplication event. The daughter species all have them. But on the lineage leading to species C, this original gene got deleted for some reason. Okay. Now your PCR amplifying this particular gene, gene 2, sorry, gene 1. Here it does not, it's not there. So what it does, it picks up this copy. So you get a completely messed up tree. This tree is basically this tree, except D is not there, and all of these are not there. So D is, oh, sorry, C. Okay, so you're getting a completely messed up tree because you didn't use the right copy. It's almost like using a different gene. Okay, so that's why people say that try to find single copy genes. So this discordance, sometimes this kind of funky relationships can arise because of use of paralogs in one of the species, one or more of the species. Okay, does that make sense? Yeah. Okay, then all right, we have done that. So in fact, what people have done is, to understand duplication events, you first get your species phylogeny using single copy genes, and then you're looking at some other gene like the globin gene cluster. There are thousands of copies, right? So you sequence all those copies from number of species. You then build the phylogeny based on gene families, right? And it's you've got the same species all over the place because there are multiple copies and these duplication events have occurred at different time periods. Then you take that phylogeny, try to reconcile it with the species phylogeny, and that will tell you how many duplication events have taken place and where, right? So there has been one duplication event here, resulting in these two clusters of the same set of species. There has been one duplication event here, resulting in that. There has been one duplication event here, resulting in that. Okay. So here's an example of a gene called interleukin, which again has many copies, and that's the species phylogeny, and then you try to reconcile, and then you come up with how many gene duplication events have taken place, right? So you can, genes that have multiple copies, you can use them as long as you're using the same copy, as long as the comparison is orthologous, okay? Here's another complication that I wanted to quickly talk to you about. Often mitochondrial regions from the mitochondria get incorporated in the nuclear genome, and these are called nuclear copies of mitochondrial genes, or NUMTs. And what happens is if you accidentally now sequence the nuclear copy, again you'll get a messed up tree. It's a huge issue with mitochondrial genes. So please be very careful while using mitochondrial genes. So there has been a nuclear transfer event from mitochondrial to nuclear DNA here. So now basically it is like a duplication event, except that for every mitochondrial gene you have a nuclear counterpart, and in the nucleus these genes have basically lost the function. They are non-functional. They are pseudogenes. Okay, and so basically then you'll get two clusters just like here, right? And again, if for some species you use the nuclear copy, and if there's a mix of mitochondrial and nuclear copies in your alignment, the tree will be messed up. Yeah. Vivek, yeah.
It would be a problem that if they are in the nuclear region, and if they are not really coding for anything, They would not evolve the same way they would in a...
Yeah. So that's why the branch lengths are shorter for the nuclear copies. So they are not evolving at the mitochondrial rate. And the other way you can, so the phylogeny will look very strange, right? You have this one species, you have actually got the nuclear copy for that one species. It will be sister to everything else. And you're like, that does not make sense, right? And because it's a pseudogene, it will have indels and stop codons often. To avoid this, you can also do something called long PCR. You try to get the complete mitochondrial genome. You're more likely to then pick up the mitochondrial copy. Or you can do RT-PCR. This is not to detect COVID, but what you can do is extract RNA from the organism of interest, and for the mitochondrial gene, do RT-PCR, get the cDNA and sequence that. This is to ensure, because once it is transferred to the nuclear genome, it's a pseudogene, so it is not being expressed. So if you pick the mitochondrial mRNA and get your cDNA from that, you have picked the mitochondrial copy. Okay, but that's in the extreme case. I think you can identify them using this and then go for long PCR. Just try to sequence a larger chunk. It's very unlikely. In some species, the complete genomes have been incorporated into the nuclear genome. A complete mitochondrial genome has been incorporated and then duplicated. Right, so it's a nightmare. So you need to check, whether in your system there's any complication of that kind or not. I believe in plants also you have this case, right, where even the chloroplast genes have moved into nuclear, some of them. Do you have nuclear copies of chloroplast?
I don't have any example yet.
Okay.
But theoretically it should be possible.
Yeah.
Many of the chloroplast genes have moved into the genome. But they're lost in chloroplast.
Oh, they're lost in chloroplast. So the same thing has happened with the mitochondria, right? Many of the mitochondrial genes have actually moved into the nucleus and they are functional. And they have been deleted from the mitochondrial. I'm not talking about that. I'm talking about a situation where, the genes still exist and are functional in the mitochondria. And they have a pseudogene copy in the nucleus. Okay, so that can really complicate your story. So in fact, in langurs we found many such cases, right? So these are all the nuclear copies of the mitochondrial genes. So the actual phylogeny should contain just that and that. And if you add in all these things, it just messes up. So it took us a while to figure out which was the mitochondrial copy. So just keep that in mind when you're using mitochondrial DNA. Okay, so have you heard this term, percent homology? Do you guys use that term? Don't say percent homology. Yes, either two regions are homologous or they are not homologous, right? Percent similarity, that term you can use, and you can say, okay, percent similarity, because similarity can also be because of chance. By chance alone, two regions can have 25% similarity, right? But the similarity, as the similarity goes up, you're like, hey, wait a minute, they might be homologous. So percent similarity can be used to say that they're homologous or not, but you can't say percent homology. A lot of people in doing molecular biology tend to misuse this term. So please don't use percent homology. It's almost like saying percent brotherness or percent sisterness. Someone is your sister or brother, or he or she is not. Right? And you determine that by percent similarity. How many, the degree of relatedness is 0.5. Oh, she's my sister, or she's my brother, right? So you don't say the percent sisterness is 50%, or it's 50 or something like that, right? It does not make sense. but people love to, take big words without understanding them, and then add on something stupid to it, and they think they are being very intelligent. So don't use that. Homology use it, but don't. And percent is also fine, but don't use them together. Okay. Any questions so far? I'll just move on to frequently asked questions after this. So I hope you're able to appreciate the fact that building phylogenies, interpreting them, it's not as simple as just sequence something and shove it into some program. It spits out some tree, and then you're like, hey, I've done phylogenetic analysis, right? You have to make sure you're using the right kind of markers, make sure all these issues are taken care of, and outgroup issues and all that stuff, right? And then only then will you be able to get a robust phylogeny, and you should be able to interpret it. Okay. So I'll quickly go over some of the. If there are no questions, let me go over the favorite part of this workshop, I guess. I'm going to go over some of the questions that people have asked us over the last 10 years. And if after that you still have some questions, let us know. Okay? All right. So I talked about all of this. Well, I have not talked about taxon sampling yet. So there are all these things. We talked about choice of markers, homologous gene comparisons, nuclear copies, gene tree versus species trees, conflicts that can arise, between across different trees, genes. Then we talked about whether we should combine the data set, look at it separately, and all that, right? Okay. The first question that that is often asked by people who are building phylogenies is how many species should I have in my phylogeny? Okay, taxon sampling is very, very important to get a robust and informative phylogeny. So let me give you an example. Here is a genus, and it has 9 species across the world. Okay, in India you have 1, 2, and 3 species. Okay, if you sample all of them, this is the tree you get. There's one species, A3 in India, which is related to species in Southeast Asia. A4 is also related to species in Southeast Asia, but they are not, A3 and A4 are not sister. So there's been 2 separate, maybe, dispersals into India, right? And then there is another species, A7, which is actually related to species in Africa. So it's a very different evolutionary relationships each of them have. Okay, you're studying this particular genus in India, and you sample in India, and you build a tree. This is what you will get. All that the phylogeny is telling you is A3 and A4 are more closely related to each other when compared to A7. A7 is more distantly related. That's about it. It is uninformative. In the larger global phylogenetic sense, where does these species fall? No information. Where could they have come from? No information. Right? So many of us are doing this. So try to avoid doing stuff like this because it's pretty useless. I mean, you won't be able to publish it. So there are 2 ways out of it, right? One is you pick a taxa for which this part of the work has been done, and you fill in the Indian taxa. when I say it's done in the sense that at least some sequences are available. You can't get all the species. My sort of rule of thumb is at least 70% of the species in the taxonomic group that you are looking at, you should try to sample. You can never get 100%, right? So that one approach is that. So you look for groups for which their sequence is already available. Secondly, collaborate with people who have samples from those regions and ask them to do the sequencing there for those groups, and so both the approaches can be done. Sorry, you had a question?
Yeah, so this is a wrong case, but in case we have a bigger phylogeny and I know what is actual sister relation. I need not to give, because it's a very lengthy phylogeny, I need not to give it. I don't want to give the entire one for my work. I just want to show the sister relation of a specific clade. So if I use only limited data, knowing the entire phylogeny, and if it's giving the same thing, then it's fine, no?
Yeah. If you're, for example, if this relationship is known, and you are now sampling more of A3, maybe there are some cryptic species there, you're sampling more of A3 and you want to build a phylogeny, and you include only these two species, that's good enough. That's okay. So a lot of times it all depends on what is the question you're addressing, your sampling strategy, right? But I come across such sad cases from India. I don't know whether to cry or laugh. There was a paper that was submitted to Current Science where this kid in some state, let's not name the state, had done phylogenetic analysis of spiders from that state only, right? So his project, I'm sure his PI was this extremely brilliant phylogeneticist the world has ever known. So he must have told him, I want you to build the phylogeny of this spider group for our state. So look, there are all these sequences available for this group. So what he did, this poor kid, all these sequences are available. He just picked up the sequences from that state, of species found in that state, and built a phylogeny. Okay, and was very proud, and sent it to a journal for publication. And I was just looking at it and thinking, hey, wait a minute, this is a very specious group. Why only so many species? And it's a phylogeny of some spider group from that state. And then I realized that this student had downloaded the sequences from another that was published in another paper that was like global phylogeny of that spider group. Right? So it's really sad. I don't blame the student. I blame that brilliant Nobel laureate of a PI who should be kicked out of that job. Another example. This happens very often, again, thanks to our wonderful PIs. Someone is working on some group, let me name the state here, so it's okay. So let's say Karnataka. Someone is working on, it's a group that is, say, endemic to India or endemic to Western Ghats. Some frog group. And this kid is told, okay, you build the phylogeny of this frog group from Karnataka. So what the kid does, goes samples, border Kerala, he turns around. He sees another species there. He's like, oh, my wonderful Nobel laureate PI said, only Karnataka. We are Kannadigas. We have to be very proud of our frogs. Why should we build a phylogeny for the Malayalis? Right? Or the Maharashtrians? I'm just exaggerating here, okay? So the kid just collects in Karnataka. This damn frog is found throughout Western Ghats. He or she can get a complete phylogeny of that group and do a wonderful job, right? But no, it's getting so ridiculous. Some districts, right? There are Okay, I don't want to name names here. There are some jokers in the field who are doing such brilliant work, and I feel I really cry for the students, you know. So
Recently, one impressions group also.
they have done the same thing.
Kerala.
Okay. By the way, every state has morons like that, so we're not targeting any particular state.
No, the problem is the PhD student would have finished all his or her work. Then
Yeah.
going for publication, it won't get published.
Yeah, it won't get published.
Time gets wasted.
Yeah. Okay, I think I should name this wonderful worker. All the papers he publishes, his wife and his kid is also on it. Okay, some of you know this guy. they do stuff like, you know, phylogeny or some group from one particular district. And his last name is Lou. So it's Lou and Lou. Okay? Oh man, such jokers. Okay, so try to sample at least, you know, 60 to 70% of all the species. And again, it really depends on your question. So sometimes a lot of sampling is probably not needed. Right?
I have a question like for
Yeah.
character evolution.
For what?
Character evolution.
Character evolution.
Like we are doing like globally, and for some group, very less information is available. How do you tackle?
Yeah, that is a big problem. You can code it as missing data, but depending on what method you're using, then that missing data is interpreted in a certain way by the program. That's not a good idea. Try to get as much information as possible. Janvi yesterday must have mentioned, right? So there's missing data in terms of the character, and there could be missing data in terms of taxa, right? There are, I think, statistical methods to try and correct for it. So where, you know, you include the taxa, but you don't know the phylogenetic position. So for example, you might, so you have this phylogeny, but you don't have, say, that species and that species in the phylogeny, and you want to do character evolution. You know the character state because that has been published, but you don't have that species in the phylogeny. So what you do is you place that species in different parts of the phylogeny and do your character evolution. And you try and see if you get a different result than when those species are not in the phylogeny, right? So there are ways to sort of get around it, but try to get as much information as possible. Okay, and sometimes what also happens is when you drop a particular species. So, for example, this is the complete phylogeny. Here, this particular species has been dropped, A6. Okay, so when you drop A6, A5 should still be sister to all of this, right? Instead, what happens is because these branch lengths are very short, A5 becomes sister to this. So incomplete taxon sampling can sometimes give you erroneous trees if it's a rapid radiation and, you know, these branch lengths are very short. Okay, so it's important to have as complete taxon sampling as possible. Okay, how many markers to use? This is another very commonly asked question. A single gene is no longer sort of publishable. You have to have multiple genes. A single gene is such a small, tiny part of the genome. It often might not give you the right phylogeny. And then we talked about how, you know, gene trees can differ from each other, right? So you need confirmation from multiple genes that this is indeed the true phylogeny. So it's nice to have many genes. Complete genome would be the ideal situation, but it's an overkill. Getting the complete genome and, you know, trying to build phylogeny. You know, you'll have to go through and get rid of all the repetitive elements and all that. It's just such a plane to do that. So it's computationally also very intensive. But what people are realizing now is that between 10 to 20 markers are adequate for most studies. And these markers are, say, about 1,000 base pairs, right? So 10,000 to 20,000 base pairs of data across the genome is usually adequate. So when do we decide whether you need to add more? We have discussed this. You look at the bootstrap support. If the bootstrap supports are very low, then you can add more data. If they continue to be very low, look at the branch length. Probably it's a rapid radiation. Use species tree approach, right?
One question.
Yeah.
Also, it should depend on the objective of our work. For example, a taxonomist working on something,
Yeah.
and he has just done some identification and want to just confirm it with molecular identity.
Yeah.
So if even with one gene he is getting more.
Oh, yeah.
So
That's good enough. Yeah, but you're assuming that there is no discordance. Say, usually what happens is there we use mitochondrial DNA. Yeah, it's okay as long as there has not been a hybridization event or anything like that. Then mitochondrial genes can give a slightly different phylogeny. Mitochondrial DNA tends to integrate more often than nuclear. But yeah, that's absolutely valid. That's absolutely fine. Okay, and yeah, we talked about this nuclear versus mitochondrial. Just keep in mind that haploid, rapidly evolving, 1/4 the effective population size. Similarly, Y chromosome versus autosomal, you can get differences. And we also talked about neutral markers versus markers under selection. Ideally use neutral markers, but none of us actually use neutral markers. We are all using genes. As long as they are under negative selection, it's fine.
what are the examples of neutral markers?
Neutral markers would be, say, intronic regions. Or what people call junk DNA. So intronic regions might also be under a little bit of background selection because they are, on either sides they have exons. But the actual coding region of the genome is really very tiny, right? They're about, for example, in humans, we have about I don't know what's the estimate now, 20,000 to 40,000 genes or something like that, right? But human, so that would be, I don't know how many base pairs, a very tiny proportion of the actual size of the human genome. The size of the human genome is more like, what, 2.5 billion or something like that, no? So 3 billion base pairs. And this actual coding region is 1%. So the rest of it is all, some of it might have some function, and then you have just random sequences that are probably evolving at a neutral rate. So I think in future, once we have complete genomes of many organisms and they have been annotated, then we'll be able to pick out the neutral regions and use that, right? Yeah, so if we are building a phylogeny, it's nice to use a whole different classes of markers. Mitochondrial, Y chromosome, and autosomal. And I wouldn't use more than one mitochondrial gene or more than one Y chromosome gene because they are all linked. They'll give the same exact story, right? And autosomal, I would use more, maybe 10, right? And if you're looking at higher level relationship, I wouldn't use mitochondrial at all, right? Okay, we also talked about whether we should combine data set, the different data set, or by concatenating the genes, or you should look at them separately. We already talked about all these things, so I'm not going to go over it. Yeah, often mitochondrial and nuclear topologies are quite different, and when you combine them, the concatenated, the tree based on concatenated analysis looks more like a mitochondrial tree. So it's highly biased towards mitochondrial tree. This happens all the time, right? So I would keep them separate. Okay. And of course, one can do ILD test or partition homogeneity test to see if the signals are different. You look at the tree topology, you can do ILD to figure out if the signals are different. If they are very different, keep the two trees separate, right? Okay. Yeah, this is the same thing. 4 approaches, combined data set. If the gene trees if there is no conflict, you can also build consensus trees. I'll come to that in a minute. Show individual trees if they are very different. Show them. You don't have to show just one tree, or use species tree approach. If the branch lengths between speciation events are very short, then probably that would be your best approach. Okay, so consensus tree. So here is a phylogeny, 3 different phylogenies that have been built using 3 different genes. Okay, and if you look at these 3 phylogenies, the higher level relationships are the same. The difference is here. The higher level relationships are fine, right? So if you are interested in the higher level relationships, you should not bother, you can combine it, right? So you can just, yeah, but if the higher level relationship is different in the 2 topologies, but you are interested in the relationships within these clades, and those are not different, then you can show the higher level relationship as a trichotomy, and just show the relationships within these clades, right? So it all depends on your question. So discordance may not be a problem all the time. So here's another example where there is possibility of hybridization. Either you show it as a trichotomy, but if there is good evidence that it's hybridization, you can show it like that. Okay. How many samples per species? Now, if the taxonomy of that group has been well worked out, just 1 sample per species is enough. Because intraspecific variation, that is variation within species, is much lower than interspecific variation, right? So if you use multiple individuals, they'll all cluster like this. In the initial days, people were asking questions like, oh, what if you use a different sample, maybe for a particular species, maybe you'll get a different tree. That does not make sense. I mean, it's like saying if you use my DNA, humans and chimps will be sister. If we use someone else's DNA, human and gorilla will be sister. It does not make sense, right? 2 human beings are more closely related to each other than either of them is to other species. So that's never an issue. However, if you suspect that the group that you're working on has many taxonomic issues, cryptic species, what have you, then you have to sample more extensively. This is particularly true with widely distributed species. Okay, and the example I often give is the Hanuman langurs, right? It's a widely distributed, this is all considered a single species in the past, found from Himalayas all the way to Sri Lanka, right? So this is not from Hanuman langur from North India, this is Hanuman langur from South India, this is Hanuman langur from Sri Lanka. In the mitochondrial tree, they are all over the place, right? If I had used Hanuman langur from Sri Lanka, and these are 2 other species of langurs, so if I had used only Hanuman langur from Sri Lanka, this is the phylogeny I would get. If I had used the langurs from India, this is the phylogeny I would get. But this species is widely distributed. So, once you sample all of them, you realize, oh, these are all different species, right? So if there are taxonomic issues in your group, particularly widely distributed group, then you'll have to do more extensive sampling for each species. I usually, say at least have 5 individuals from across the range. And as Chaitanya was telling the other day, from the borders of the range and a few from the middle, right? So this is something you'll have to decide based on the group you're working on. Which tree building method to use? What would you recommend? ML and Bayesian. Fortunately, both almost always give the same topology. Parsimony has gone out of favor nowadays, out of fashion, because it does not use a model of sequence evolution. And
people still use NJ papers.
yeah, some papers use NJ. It's okay, as we discussed earlier, just to get sort of a feel of how species might be related. If the data set is huge, sometimes people show NJ. But ideally, these are the 2 methods one should be using. What do we do if different tree building methods generate different topology? Report both of them. I've never, between ML and Bayesian, I've never had that problem. Right? There might be some minor change at the tip, and that does not really matter. So you just mentioned that, okay, the 2 topologies were different with respect to this one species, which moved, instead of being sister to A, it was sister to C or something like that. But overall, that change does not affect the conclusions of our paper. So we are just showing either ML or Bayesian, right? But if the differences are significant, then you should ideally show both the trees. But I've never faced that problem. So where is the incongruence? Try to figure that out. If it is important, then you report it. Maybe more data will again resolve this issue. So that is something to keep in mind. If one or more species in my tree is not monophyletic, someone showed me a phylogeny the other day where, the species that she's looking at is all over the tree. It's polyphyletic. So there are all kinds of, if it is closely related group, then it's probably retention of ancestral polymorphism, incomplete lineage sorting. But often when you have polyphyly across a huge phylogeny and similar looking organisms all over the place, it's convergence. You need to revise the taxonomy. And of course, hybridization often can result in a situation that looks which results in groups not being monophyletic. Yeah. Okay. So those are some of the questions. I think how do I treat gaps? So if you have gaps in your data set, you code them as question mark, and program interprets it as missing data. Now, there are 2 ways of dealing with gaps, and it's important that you understand the difference. Is the gap a true gap or not? Is the first question that you should ask yourself. Right? What is a true gap? Well, you have a gene sequence from one particular species, it is say 1,000 base pairs, it's a coding region. In another species, there have been a couple of amino acid deletions, and that has introduced a gap in that species. Okay? Now that is a true gap. It is not missing data in the sense that when you were sequencing, you were not able to get that region. That sequence does not exist. Right? So that's a true gap. Now, in this case, ideally you should not treat it as missing data. you should just get rid of the whole thing across all the species. Why should you treat this as missing data? Because what is missing data? Missing data is, you have maybe you're using 3 genes. For some species, you have all the 3 genes. For other species, you manage to only sequence 2 genes. This is true missing data. That data can be generated, but you didn't have the time or the money, or the PCRs were not working. This is truly missing data. Whereas this is a true gap. Right? So when you treat it as missing data, likelihood and Bayesian approach will say N. N means it can be A, T, G, or C. Here it's not N. It does not exist there. Right? And the program deals with this. Remember, we were calculating likelihood, and at the tip there was, say, A here and a T here. Now, while calculating the likelihood, now what it will do, it will have A up here, and it will have A, T, G, and C for the other terminal node. And it will go on to calculate. The computation goes up. Right? So that is what N is. So if you have not sequenced it, you can code it as missing data. But if it is a true gap, then you have to basically drop all those characters, ideally. Don't treat it as missing data.
This we are
Okay.
talking about single gene or
Yeah, this is single gene within a gene, right? However, you can also have a situation where this this gap is actually informative. It is telling you that these 3 species are more closely related because they share a gap, right? So then likelihood, unfortunately, you can't use that information. In Bayesian, you can introduce this as a separate character. You get rid of this part, and then you use this as a separate character where these individuals don't have a deletion, but there's a deletion here. And there are ways to incorporate this in Bayesian analysis. In parsimony also, you can use this as a 5th character. But in likelihood, you can't. Yeah.
Sometimes we might have a problem identifying the true gap, right? For example, say I'm looking at a gene A. Some people outside the country have sequenced that gene for multiple taxa. They have got a sequence length of say 1000 nucleotides. I have tried to sequence the same gene, and I have got somewhere around 900 or 850 bases.
Yeah.
But while sequencing, while checking the chromatogram, we can check that whether I have been able to completely sequence or not.
Yeah.
So at that time, we cannot trade if my final whatever sequence.
Yeah. So then it's missing data.
So just treat it as missing.
Then it's missing data, right? So you're saying the published sequence is that, and you're not getting the complete. Maybe on one side you're getting only so much, and you're confident that it is not because if you get to the end, you'll probably see a stop codon, right? And till here it's quite nice, and you're not able to get this. So this will be missing data. This is fine. So you can do n so or question mark. If you put question mark, the program will treat it as n, right? But I hope you understand that this is not n, right? This is a true gap, or it can be an insertion. It may be an insertion here that would introduce gap here, right? So that's how you treat with partial sequences. Gaps. And partial sequence also same thing. You treat it as gap. Question mark here. Sorry. Yeah.
Can you do a pairwise deletion instead of coding separately? Can you do a pairwise deletion instead of coding?
No, pairwise deletion would come in case of genetic distance. So when you're building distance-based tree building methods, so that time you can do pairwise deletion. But for likelihood and Bayesian, it does not make sense. You know?
And what are the chances that if I'm lining a gene, suppose it's a protein coding,
Huh.
ideally there should not be any gaps in between the coding region.
Huh.
If there are any gaps in my alignment, what are the chances that it's a sequencing error that
No, you can have gaps. As I said, even in a protein coding gene, in species A, you know, it has 150 amino acid. Species B has, 140 amino acids. There, 5 amino acids have been deleted. So as long as the deletion does not cause a frame shift mutation, it's fine.
In coding regions, the deletions will be 3-3.
3-3.
Easily you can identify. Insertion or deletion will be in 3.
3.
Triplets. So you can identify whether it's a real one or an artifact.
Yeah.
If 2 nucleotides are missing, then sequencing error. If 3 are missing, it's more likely it's a deletion.
Deletion. So if it is just one missing nucleotide or 2, that will cause frame shift mutation. When you translate it, immediately you'll know that it's a problem, or it's a pseudogene. You might have picked a pseudogene. Pseudogenes can have all these changes, right? But a true deletion, as Siddharthan pointed out, it will be in triplets, right? And we see this all the time in the data set that is there in your practicals, protomine. There are many amino acid deletions, some of them 3, just 1, so it happens. Okay, do you guys have any questions? These were the frequently asked questions that I could come up with. The most important thing is taxon sampling. Hey, is the tea here? Okay. So if there are no questions, let's break for tea, and then 11:30, Siddharthan will give his talk. So let me stop this.